and thanks everybody for joining us today. We have some exciting new features in SeqBuilder that I can demonstrate to you. So we'll start with a few slides here just to kind of introduce you to some of these new features, and then we'll spend most of the webinar in the software taking a look at them live time. So SeqBuilder is part of the molecular biology suite at DNASTAR. So we're really going to focus on this one program. There are other components to the molecular biology suite, and they provide a wide-ranging function ranging from multiple sequence alignment and phylogenetic alignments to ABI trace file alignment. And you can see we have a picture of a circular DNA molecule here. So we have a nice new interface in SeqBuilder for visualizing sequences and doing different analysis. So we'll really be focusing on that component today. And of course, we've been developing new functionality for SeqBuilder for over a year now. So we have a nice list of things that we can go through. And so some of these new features include a new interface that's highly flexible and customizable, and that was introduced last fall in version 15.0. And this new interface has different ways that you can configure the views. There's side panels. There's a dynamic enzyme label placement. We can move features around. We can do things in mass. I can change colors of features by making some quick selections. So it's this nice interface, and now we're adding some additional functionality to make the software more powerful, and also to integrate it with some of the other tools that we have in our suite. So we have a 64-bit version that we've added for 15.1. This is both the interface as well as what we call a data manager under the handle data. And so we can load as much as over 1,000 bacterial genomes into a project or an entire human genome. So we can really start to stretch the capacity, and we'll start to build some additional tools to handle this kind of scaling up of sequence numbers and sequence sizes. What we're going to focus on today are these last two bullet points, and that's the automated clone verification and an interpretation that we provide. And the way that this works is that we'll make clones or import clone sequences, and then also bring in ABI files, trace data, that we would use to verify the integrity of that clone. And it's going to use our extremely powerful SeqBuilder Pro software, kind of on a seamless under-the-hood connection, run the alignment, and then give you an interpretation of what's happened based on the alignment of those reads. So we're leveraging one of our most powerful pieces of software to align this trace data and then give you feedback as a user as to the integrity of your clone. We also have implemented some SeqNinja tools. So there's another application in our molecular biology suite that is, it allows you to make scripts and do some automation on sets of sequences. And what we're doing now is making some of that more accessible to folks who aren't comfortable with scripting, and also make that power available through SeqBuilder. So if you have a set of sequences, you can do things like edit them in batch, or extract features, or concatenate, that sort of thing. And that's again giving you access to another tool through the SeqBuilder interface. And that's a trend that I think you'll continue to see from DNASTAR. So the 64-bit SeqBuilder Pro, again, it allows us to open much larger sequences, large numbers of sequences. It will keep us compatible with the newer Mac operating systems. And, there's numerous different performance improvements that will benefit from this. So it's a move in a direction to make the software more powerful. The clone verification and interpretation, it actually means quite a bit. There's when you make clones, in some cases you just want to know, is the insert what you think it is? And you just do a quick check to see that it matches up your read. You might also have orientation. Certain cloning methods, an insert can go in one way or the other. And you can just validate which direction the insert is in. And then, of course, variations can be introduced, or frame shifts that could affect a translation, or things like a fusion protein that might be generated. And then you can look for things like coverage across the insert. Are you validating the entire clone, or from across the entire insert? Do you want sequence data? And then any reads that don't align. And you take all this information together and interpret it And of course, you can do this with SeqMan right now. I can align reads to using SeqMan, but the interpretation can be challenging. Depending on how the reads align and where there's mismatches, those can mean different things. So we've built this feedback mechanism now, so that when we do the alignment, it will give us this feedback, so we can better understand any potential problems we have with clones that we've generated. And the SeqNinja tools, here's the first set: concatenate, extract features, batch translate, batch reverse complement. And we'll just look at those as well, and the interface that we have to use those. And of course, all this information, we have a new web page for SeqBuilder Pro. So please feel free to go to SeqBuilderPro.com, and you can get some more information about this new software. So at this point, I'm going to jump out and launch the software. And I thought what we'd start with is one of the data sets that was a little bit problematic, where we had a clone and the vector was unknown. And so we make a best guess, and we can look at the result and interpret it to say, well, maybe we don't have the right vector. So I'm going to show you a clone verification starting with an actual cloning project. So we're going to start a gateway entry clone, and I'm going to import the insert sequence. So we just have this FASTA file. It's the insert. It's NOX2 gene, and it's just a simple cloning where we have one insert. Oops. Bring that in, and in this particular case, if you make a mistake and don't load the correct vector in, which is a common, where you don't quite know what the vector is, it's a pretty common. I'll make this window a little bit bigger here. Okay, so that is a simple, we'll try this out, and we get this little summary. So again, this is the cloning wizard that is in SeqBuilder Pro. So I see my vector and insert. It tells me how big it's going to be, and then what's happening here is that we're getting fragment junctions displayed, so we can kind of see what we're amplifying and what primers we need to design. So that's all part of, again, the cloning wizard. And we're just going to make this clone. And so now what we get is, if you look over here at the project tree in the left pane, we have our insert, our vector, and we have our clone that we made. So we have this entry clone, and we have the insert sequence that was also constructed from the PCR. And it immediately brings me to a clone verification step. So it recognizes that we just ran a cloning experiment, and it's asking me, do I want to validate this clone with some ABI reads? And so I can drag and drop them in, or use this browse button to bring in this case, it's just a simple forward and reverse ABI read. So I bring them in, and you can see in this little picture in the upper right corner, it gives me the size, it gives me a landmark of the origin. There's a little tick mark there, and also some numbers to show me at what position this insert is placed in the clone. And so when I verify this, I want to know, is that insert in the right way? Do these reads, I expect them to match, across if I've designed this a certain way at the size, it should match across the entire insert. So when I select run, what's happening now is it's actually sending the sequences to SeqMan Pro, our one of our assembly algorithms. And it's aligning those ABI files to the clone. And immediately, just a couple of seconds, I get some feedback. And this is a clone verification summary. And I get a picture with a kind of a statement of what happened. And it just says that we have a clone confirmed. And I can see the reads there, a green and a red read. And they're labeled a forward and reverse. So they both aligned. And it looks just from the picture that, okay, they aligned in the right spot. So I'm fairly satisfied. But then there's some text here that says there may be some mismatches that I want to examine. So these reads align, but there are mismatches. And in this particular case, what we have now is essentially a variant table under notable variations. And as I scroll through, I can see, oh, there's actually quite a few mismatches here. Even though the reads aligned overall, there are some stretches just here where we have mismatch. And the columns then have different bits of information. We have the position within the clone sequence, and then we also have an alignment position. And these are links then that either bring me to the sequence file or it'll bring me to the alignment position. And it tells me, is this variation occurring in the vector sequence or the location, or is it in the insert sequence? And then I get information as to how far from the insertion site is this variation occurring. I may not be as interested in variations that occur a long ways from the insertion site. And I see these are kind of in a string, so I want to look at this alignment now. I need to interpret what's going on here. And so I'm going to select this 1119 position. So I click on the link, and it's going to launch our SeqMan Pro software, which is an excellent piece of software for evaluating trace data. And so we'll expand this a little bit here, give ourselves some more real estate. And what it's done now is it's loaded the alignment then into SeqMan Pro, and SeqMan Pro has this interface that lets me look at the ABI trace files, and this is a fully functional, I own SeqMan, it's licensed, fully functional SeqMan Pro, and I can see some blue highlighted residues here, and these residues indicate differences between the aligned sequence data and my clone. So I see this string right here, and the twisty triangles here allow me to expand the trace file, and also on the clone, the annotations that are present. I can make quality scores visible here, right? I can do trimming. I mean, SeqMan's an incredibly powerful program. I can scroll through. So here's the end of the trace file. I can extend the end and see some of the ends that were trimmed off. So I can look at this and I see, well, it looks like it matches, so clearly the read matches, but there's a string and it's right at this ATT site. And so what that probably means is that when I made my in silico clone, I may have picked the wrong entry clone in my gateway that had a different ATT site, and I can kind of identify that. So I might go back and say, well, I need to reconstruct my in silico clone to match what I actually had in the lab. And I'm just going to scroll through now, and you can see different positions in this file that have mismatches. And now here's the reverse read coming in, and I can extend the trimmed end here and see some of the trim. You can see there's the reverse read, and I'm just kind of scrolling through, and you can see, oh, there's another site here that has a similar. So even though there's lots of variations, they're all mostly in a string, and it has to do with differences in the clone. I just selected the wrong clone. I can also navigate this alignment using the SeqMan SNP table. And this is a fully fledged SNP table, and so I can select a region, and I'm just going to kind of use my arrow key, and I can very quickly move through the alignment just by arrowing through the SNP table. It's very responsive. So I can quickly evaluate the alignment and decide what's going on with this particular sequence. Okay, so that is some real data where there was a mistake made just with the clone that was selected for the virtual clone. So let's go back to and show a different kind of a scenario. And I'll close this so we're not. Okay, so now I'm just going to open a clone sequence that's already been made. So you may have clones that have been constructed, and I'll just show you one that has. So here's a sequence, a clone that was constructed, and it has an insert sequence. And if I double click on this, so this again is the new interface, it will bring me to, let's make this a little larger, I think, the feature table. And you can see there's an insert feature that's been created there. So this particular clone was made in SeqBuilder, so it has an insert feature. So if it has an insert feature, I can now go to cloning and say, I have data to verify this clone. And I'm going to bring in some Sanger reads again, run it. And here's what we hope to see most of the time is we have a clone confirmed, no significant variations are reported, and essentially we're good, right? And we can either save it or just cancel out of it, depending on what our particular need is. So that's what we hope to see most of the time. Now I'm going to show you a couple scenarios here where we have some issues. So I'm going to show one that is a clone that maybe came from a different source, and it doesn't have a recognizable insert feature. So what I did is I replaced that insert feature with, changed the feature type essentially and made it a MISC feature. So there's no identifiable insert here. So if I try to verify this clone, it gives me a set insert location button becomes active. And it won't let me run. It's not going to have the information that it needs. And you can see my picture here where it says choose some reads doesn't show that there's a recognizable insert with this sequence. So I click the button to set the insert location, and it will bring up that sequence in a viewer, and it's going to ask me to select what the insert is. So I can either click on the feature to select the range or select a range. So now I'm telling it that that's where the insert is. So you can specify the insert, and now again it's same kind of process. Select a couple reads, run, and we get the same result. So again, if it's any kind of clone data that you receive from a different source where you may not have an insert feature, you could of course go and create one ahead of time, or just use the cloning wizard to allow you or the verification wizard to allow you to specify that. So now let's bring up a different scenario. So here's one where the insert is reversed orientation. So same sequence, this time we have the insert in a different orientation. Use the same set of reads here, run. And now we get some feedback that says, hey, we've got a problem with this clone. And when we look at it, we can see these kind of pink wedges. And these highlighted regions, what it's saying is that there's regions here that have a high density of mismatch. And it's right at the place where the insert is inserted, right at the insertion site. And so it's telling the user, this disagrees, and we likely have a problem here. And of course, that when you see something like that, that is likely, doesn't always mean that it's a reverse insert, but it is very like a reverse orientation on the insert. And so when I go to the variations now, you can see there's not that many SNP variations, but now we have notable conflicting regions. So it's these wedged, pink wedge areas, and we can click on the link. It will bring us to now a strategy map in SeqBuilder Pro. And so the strategy map has a histogram of conflicts. So when there's so many that you really can't display them separately very easily, it just plots the conflicts in a graph. And it just kind of confirms that, yeah, we've got a lot of disagreement here. If I double click on that map, now I can go and look at the aligned reads and identify areas that have lots and lots of conflict. And so there's, so again, that's an interpretation then that says, you know, we've got a problem, and you're going to want to look at the alignment and validate what might be the problem. Okay, so that's another scenario. I'll just show you just one more here. So we'll open up something that's a little bit bigger now. So we've got a, where is it here? Larger insert sequence. So now we have a sequence that's got a large insert in it. So there's going to be more ABI data. We're going to try to verify the entire insert. So we have multiple layers of ABI sequence. So we'll verify this clone. And now we have a little bit larger set. So now you can see our map is different. We've got quite a few more reads. They all match, so we're just confirming in this case that we've got reads coverage kind of across the insert, and we've confirmed orientation, so everything looks good there. Now I'm going to actually save this one. And so here's the saved clone, and we can see I've turned off the other features to make these pop out a little bit more, but now the saved sequence takes the ABI files and automatically converts those. If I go to the feature list here, makes a feature type called a layout, just a layout feature of where that ABI file aligned. So I get features created automatically then for that particular sequence. And I'm going to give ourselves a little more real estate here. And we also get a history, so we can change the view here. So we have, and we save these steps then to the history step. So we have this, and this can be, there's different ways we can even move this out. So I can export it as HTML or bring this to a lab notebook or something if you want these images somewhere to save them. And so again, large insert, we get nice coverage. We can do that alignment, no problem. So that's the different steps. So again, different scenarios where we have lots of variations, reversed inserts, other kinds of issues. This feedback then, when we do this verification, will tell us that we've got these sorts of issues. Okay, so I'll close these out. Now we'll take a look at, so that's clone verification. We'll take a look now at some of the new functionalities.
Oh, hi. This is Sharon, the host. I'm just going to interrupt Matt for a second because we actually have 4 questions regarding clone verification, and I thought it might be nice to go ahead and deal with those now before we go on to the other features in SeqBuilder Pro.
Sure, absolutely.
Okay, so the first question is, do I need to own SeqMan Pro in order to have access to the clone verification?
No, you don't have to own SeqMan Pro. So if you don't own SeqMan Pro, you'll still be able to do the clone verification alignment and visualize all the output. The only thing that you won't be able to do is edit or add more sequences to the project.
Great. Next question is there a limit to the size of clones that can be verified?
SeqMan Pro is a powerful aligner, so it can handle thousands of ABI files, so that should handle any size clone that you might have.
Terrific. The next question is, do you need to have the insert data as part of the clone sequence, or if you just have the completed clone sequence, is that enough to be able to do clone verification?
Yeah, what I showed in the demonstration is that you can specify the insert in the clone verification wizard, or you can open a sequence and create an insert sequence anywhere, and it will read that and use that as the location.
All right, and then here's the final question, which is, so this user says that he has batches of intended clone sequences and then batches of reads. Can he do clone verification in batch mode?
We do not have batch mode at this point, but what I'm going to show you now some of the batch mode tools that we're developing, and that will be something that is added in the future. So yeah, that's the direction that we're working in right now.
Great, thanks, Matt. So I'll go ahead and let you return to your presentation. Thanks. Okay,
thank you. All right, so that's a good segue then to the batching tools. So you can see that in SeqBuilder version 15.1, there's a tools menu, and now we offer some automated tools here. So when I select one of the tools, we get a project file window again, and I can import data in. So I'll just bring in I have some GenBank sequences here that have that are fairly good size and have lots of different kinds of annotations added to them. And so, but I didn't select the right one, so I'll move them into my sequences folder. And so once I've loaded sequences in, I can select one or all the sequences. In this case, we'll select the set and name it. And I can pick the output. And you'll notice here that I may have FASTA files, and if I want to keep track of the segments, I can create a source feature then for them. In that case, we'll want to use an annotated file. These particular sequences have source files already associated with them, so I won't check that box. And we get a little statement here that says how many sequences will be concatenated. And what's happening here is that this is going to actually write a instruction to our SeqNinja program. And so it's actually writing a SeqNinja script. And of course, for advanced users or folks comfortable doing scripting, there's more capability built in SeqNinja. We're just making some capability accessible via SeqBuilder here. And so I'll run this concatenation, and now we get a concatenated file. And I can take a look at that file, and it will open this up with all its features. And we get, as I said, a feature with a concatenated sequence with lots and lots of features. And so again, some functionality that is new to 15, but not 15.1, is if I decide that's too crowded, I can go into this list, the feature list. And for example, if I want to make the STS features go invisible so I don't see them, I can select them, then select all of that type and hide. So I've just hidden. So let's just, again, make my, and you can see I'm just kind of zooming in and just showing you different, all the different annotations that are present here. So it's pretty loaded up with different annotations. And we can zoom out now and get a feel for the entire sequence. So that's just a concatenation now. We have these sequences together. I believe they all have sources too, so I could have, here's the source features that are not visible by default, but I could make those source features visible, or even let's see, I'm going to select it here. Let's see, how do I get that one? Oh, there it is. So there's the E. coli. So there's the three pieces there, the three different components. So those are automatically visible. So I can see I concatenated those three. So I can go back now to this folder, and I can do more. So if I want to do further steps on this concatenated sequence, I can select it and say now we want to extract features. And I can pick, in this case, I'll just pick CDS, but you can see the wizard here will look at this sequence and read the feature types and tell me how many features I have in either a concatenated sequence or a set of sequences. It'll give me a feature count by the type, so it automatically does that. You can see as I'm changing this, the number changes. And there's also a filter. So in one example, I may want to just extract all features that have a certain gene name. So if it's a whole set of plasmids, maybe all the ampicillin resistance genes, I want to compare them because there's sometimes slight differences. So it allows me to import a large set, apply a filter if I want, and then name, again, using the different fields. I can select what field I want to search and pick a name. So it gives me some nice advanced filtering capability. But I'm just going to extract some CDSs now. So now I have the set of CDSs from that concatenated sequence. And I could either stop there, or I can continue to run tools on that. So I might decide that on this set, now I want to translate them. So I can batch translate as well. And what's happening here is as I'm running these tools, we have a script that's developed, right? So we've got a history of what we've done, and we're keeping track of that, of those commands. And I'm going to run this again. And now I have translated. Nope, that's a little short one. Let's find one more interesting. So now I can have all those sequences auto-translated so I don't have to do them one at a time anymore. So again, it allows me to automate some functionality. I can bring in, larger sets of sequences, run these tools on larger sets. So again, it takes out some of the kind of monotonous things that you would have to do one sequence at a time. We can now start to batch those particular jobs. And there's one last piece is I can also do a reverse complement. In this case, I'll just do a little subset. Oop, I want a reverse complement here, so I'm just going to select and run this last little tool. And we have reverse complemented sequences as well. And I can add folders here and construct this tree if I want to have new folders. I can organize things that way as well. And so that's kind of the start again of some automation, some batch automation. We'll have more tools added. Hopefully with each version you'll see this list grow longer, and we certainly welcome any customer feedback on the types of tools that you're interested in seeing in an automated fashion in SeqBuilder. And with that, I'll thank everybody for joining us today. And if there are any questions, I'll be happy to stick around and answer any additional questions. Thank you.
Thanks, Matt. We just have one additional question about the batch editing, and that is if somebody wants to do batch editing, do they need to have a separate license for SeqNinja?
Good question. You do not need a separate license, so you can do everything from SeqBuilder just like I demonstrated, and it will access SeqNinja and run the automation.